Hello, this is Arkasha from MCAT Simplified. In this video, we're going to be discussing organelles that are found in eukaryotic cells. And remember that organelles are membrane bound, and that membrane helps to separate the processes inside these organelles from the outside, which is the cytoplasm of the cell. So the first organelle to discuss is the nucleus, and this is the sort of command center of the cell. This is where you're going to have DNA, which is the genetic storage material. And that DNA is going to be replicated here, and it's also going to be transcribed, which is the process by which RNA polymerases will use DNA to synthesize RNA. Additionally, this is the site of splicing and general mRNA modification, wherein the precursor messenger RNA is modified through splicing and 5' and 3' modification to produce the full messenger RNA, which is then transported actually from the nucleus through these little openings in the nuclear envelope called the nuclear pores, which are right here, into the cytoplasm, and that's how it gets to the rough endoplasmic reticulum, which is actually the next organelle. Now notice that the rough ER is directly adjacent to the nucleus. They're very close. And this is the rough ER because its membrane, as you can see, is studded with these protein RNA complexes called ribosomes. And the ribosomes are important for the process of translation, where an mRNA is used to synthesize proteins, okay? And ultimately, the rough ER is also going to be where you have some protein modification. However, after that, the rough ER will export the proteins it produces to another organelle, which is called the Golgi Apparatus, and we'll talk about that next. But before that, let's discuss also the sister of the rough ER, which is the smooth endoplasmic reticulum. And it's called the smooth endoplasmic reticulum because it doesn't have any ribosomes in its membrane. And it doesn't have anything to do with protein synthesis at all, in fact. Instead, it's involved with lipid synthesis of different fats, including cholesterol. And cholesterol is also used to produce steroid hormones such as testosterone and cortisol, and so this is also the site of steroid hormone production. It's also the site of detoxification of drugs and various toxins, particularly in the liver cells. And so the liver cells will detox drugs as well as alcohol, for example. And then in some cells, it's also a specialized organelle for storing calcium, such as in muscle cells. Next, we have the Golgi apparatus, and I want you to think of this as the post office of the cell. The idea is that the Golgi is going to be receiving proteins from the rough ER, which it synthesizes, at its cis face. And these proteins are going to then go through these vesicles of the Golgi one by one, and they will undergo post-translational modifications until eventually they reach the other face, which is the transface, and this one is facing the membrane of the cell, the plasma membrane. And these proteins which have been modified are packaged into these vesicles, okay, or post boxes, or mailing boxes, and they are sent to the membrane. And at the membrane, they can either remain in the membrane as proteins, or they can be exocytosed from the cell, released from the cell. And so, in summary, it receives proteins from the rough ER, it modifies them, it packages them, and it sends them off just like a post office. Next, we have two very similar looking organelles, which are the lysosome and the peroxisome. They're spherical. But these are very different in function. So the lysosome, I'll think about it as the trash and recycling centers. These contain acids, so the pH inside the lysosomes is quite low, and they also contain hydrolytic enzymes that can break down molecules. And the function of the lysosomes is to digest old organelles, as well as various cellular debris, which can be from outside the cell or inside the cell, as well as bacteria by some cells like macrophages. And then peroxisomes are sort of the detox squad, and these contain the enzyme catalase. And catalase is involved in breaking down what we call reactive oxygen species, which are usually free radicals, such as hydrogen peroxide radicals, which can actually damage cell membranes and proteins, so they're dangerous to the cell and they have to be eliminated. And then a lesser known fact about this organelle is it's the site of beta-oxidation of specifically very long-chain fatty acids. As some of you might know, beta-oxidation mostly happens in the mitochondria, but this is one exception. And lastly, we have everyone's favorite organelle, which is the mitochondria, the powerhouse of the cell. And it's the powerhouse of the cell because this is the site of oxidative phosphorylation, which is aerobic ATP production, oxygen-dependent ATP production, which is very efficient and produces a lot of ATP, okay, which is the energy currency of the cell. Now, it's also the site of the enzyme pyruvate dehydrogenase, which is important for bridging, it catalyzes an important reaction for bridging glycolysis to the citric acid cycle. And then it's also the site of the citric acid cycle, the electron transport chain, and like I said, oxidative phosphorylation, which is catalyzed by ATP synthase. Now, additionally, it's going to be the site of most beta oxidation, aka breakdown of fats, such as medium chain and long chain fatty acids. But like I said, not very long chain fatty acids. That's the peroxisomes. And then a really interesting thing about mitochondria is there is something called the endosymbiotic theory, which is the theory that a long time ago, before eukaryotic cells evolved, the precursor, the ancestor to eukaryotic cells engulfed a bacteria. And subsequently, that precursor and the bacteria inside it evolved symbiotically to produce modern-day eukaryotic cells. And the bacteria that was engulfed by that cell became the mitochondria. And some of the facts that point towards that is the mitochondria have its own DNA, which is the mitochondrial DNA, which is circular, just like bacterial DNA is. And it's actually replicated within the mitochondria, transcribed into RNA, and that RNA can be translated by mitochondrial ribosomes to produce mitochondrial proteins. Additionally, the mitochondria self-replicates in a process very similar to binary fission, which is the process by which bacterial cells replicate too. And then also, mitochondria, another fact about it is it's important for apoptosis, which is programmed cell death. And the idea is that the mitochondria needs to break down prior to apoptosis to release a protein called cytochrome C, which can then activate caspases, which are the enzymes that facilitate cell death.